Coming to the next one is the incubation of excipient for the callus production. In this case, we know before this one we already prepared one of the slant, nutrient media slant. Okay, after that we inserted one of our desired excipient part. This is one of the excipient part we are inserted, right? With the lead tissue we are taken, and see the first side is closed with the tissue opening is closed with one of the corner plate, right? And this tissue kept within the dark room. This tissue initially kept within the dark room. See here, 2 to 3 weeks we have to maintain. 2 to 3 weeks we have to maintain within the dark room, right? And daily we have to check not daily, every 3 days or every 4 days we need to check the tissue, because is there any contamination is there, is there any fungal growth is there, we need to observe. Is there any contamination is there? Definitely we need to remove the tissue from the tissue culture room, right? So there is a separate room is there within that separate room. Okay, that is the tissue culture room. Within that room, okay, proper cooling temperature is maintained. Means optimum, all optimum conditions are maintained. All optimum conditions are maintained. So within the dark room and the cooling temperature is maintained. That is the tissue culture room. So within that room, okay, we need to incubate the excipient 2 to 3 weeks. After that 2 to 3 weeks, there is a callus production takes place. We already know the callus is an undifferentiated cell mass, especially the cells are having brown color and which are involving the rapid cell division process. Remember, within this nutrient media, there is an auxins and cytokinins. Auxins and cytokinins. Both these are used in the equal ratio. Right? Equal ratio of auxins and cytokinins are used. Right? That is about the incubation process and the excipient, from the excipient callus production. Right? And production of plants from the callus. So we know this is one of the tissue. This is one of the tissue. Within the tissue, we already prepared the callus. This is one of the callus. The callus is taken out and the callus is chopped. Right? After that chopping process, see, we prepared one of the nutrient media. Within this nutrient media, there is a high concentration of cytokinins and low concentration of auxins are present. High concentration of cytokinins and low concentration of auxins are present. So within that nutrient media, if we inoculated or inserted that callus part, so within this nutrient media, there is a shoot pod production will takes place. So one of the nutrient media, shoot pod will be produced. After that, within that next nutrient media, there is a cytokinins low concentration and auxins more concentration. So within this nutrient media, roots production will take place. So already shoot pods are produced, and later the roots are produced. Right? So we need to change the nutrient media. For shoot pod production, what kind of phytohormone ratio is there? And for the root production, another kind of phytohormone ratio is required. So, organogenesis required, each organogenesis means shoot pod requires separate nutrient media and the root pod required separate nutrient media. So, by this way, from the callus we are producing the complete plants. After the production process, the next one is the acclimatization or hardening of plants. See, acclimatization or hardening is nothing but, see, the plants are very sensitive. These are very sensitive plants. if these are directly exposed to the normal environmental conditions, the plant will be dead. All the plants will be dead. So to avoid that problem, see, there is a greenhouses are present. We know in the nurseries there is a greenhouses are there. Within that greenhouses there is a green. This is one of the manual. See, whatever the nutrients are required, all these nutrients are available in this manual, right? So here this is one of the plant pot. This is one of the plant pot. So here leaves are there. The leaves are there, right? And the plant is always covered with one of the polythene bag. The plant is always covered with the polythene bag. See, this is another polythene cover. There is some pores are there within that polythene cover, and which is supporting for the exchange of the gases. Which is supporting for the exchange of the gases. See, what I am trying to tell you, whatever that the polythene bag is there, that polythene bag, especially preventing the loss of water, right? See, if the open place is there, whatever that wind and whatever that water content is present inside, maybe the plant lost the water content. So, presence of polythene bag, there is favorable conditions are maintained. Already the plant under the greenhouse. See, we know in the nurseries there is a green color shelters are there. Within that shelters, the plants are kept. And once the hardening process is completed, up to few weeks, the plants are cultivated within that shelter, right? Once the time is completed, and these are directly kept into in the normal conditions. These are kept in the normal conditions. So that is about that one of the acclimatization or hardening of plants, right? So these plants are ready to cultivate. These plants are, once the hardening process is completed, these plants are ready to cultivate, right? That is about the acclimatization or hardening of plants. And next one we will discuss about the applications of tissue culture. See, applications of tissue culture is a very important concept. Hi students, coming to the next topic is the applications of tissue culture. In that case, the very first one is the production of virus-free plants. So how the virus-free plants are produced? First, suppose one of the best example, this is one of the plant. See, the parts, these are the leaves, right? These are the leaves, and this is about the roots of the plants. The plant is growing. This is one of the high yielding plant, but I want to produce virus-free plants. So how can we produce the virus-free plants? Remember, first of all, if we take a leaf is the source for the tissue culture purpose for the callus production. Leaf is the source here. So the leaf is maybe already affected with the virus. If the leaf is affected with the virus, if we take this one as the source, the next generation plants also having the virus, right? And if we take in the stem, or if we take in the root part, or if we take in the root, so in this case, especially in these cases, already these parts are maybe affected with the viruses or the virus. Next generation also, whatever the plants which are producing by the tissue culture, next generation plants also having the viruses. Then how can we produce the virus-free plants? Whatever the tip part is there, the tip part having the meristem. The tip part having the meristem. The meristem, whatever the meristem contains the meristematic cells, and these meristematic cells are continuously undergoes the cell division process. These cells are continuously undergoes the cell division process. So there is no chance to entry of viruses into the meristematic cells. Then one of the meristematic cell or meristematic tissue or meristem part, you are taken as source. That meristem part is subjecting for the callus production. From the callus, we are producing the plants. And all these are virus-free plants. All these are virus-free plants. Because whatever that the root meristem is there, shoot meristem is there, that meristematic parts are generally not affected by the virus. Then if we are taking the meristematic cells as the source for the production of the plants, in that case we can produce the virus-free plants. Right? That is about that virus-free plants. And remember, scientifically this is already proven. Some of the plants also produce. There is a potato plant is there, and next one is the banana plant is there. Potato, banana plant is there, right? And the sugarcane, right? These three plants are already produced by the scientists. Right? That is about that virus-free plants production. And next one is the micro propagation. Micro means small. Propagation is nothing but production of plants. Propagation is nothing but cultivation of propagation of plants. Propagation is nothing but production of plants. Right? So within the small area, many number of plants are produced. See, this is one of the tissue culture lab. So within the laboratory condition, okay, thousands to lakhs of plants are produced. We know one plant leaf source is taken from the leaf, callus production takes place. From the callus, many number of plants are produced. Right? So this is nothing but micro propagation. Remember, the micro propagation is the very important process. Especially one scientist spends his lifespan, complete lifespan, produced one hybrid plant. See, if the hybrid plant, this is one of the hybrid plant. See, from this plant, okay, the scientists want to produce many number of plants. For suppose, if the sexual reproduction is done, maybe we lost the desired characters. Of this hybrid plant. Then, how can we produce many number of plants? By the tissue culture process. If we are taking the leaf part is the source. From the leaf part, callus production, many number of plants are produced. So, this kind of plants are called as soma clones. Soma means body, clones means identical. Because whatever that parental source we are taking, okay, the producing new plants also having the same characters. The first year we studied about the clones, identically same. Here soma clones, we are taking the body cells, producing the same characters. So these are called as soma clones. Right? That is about the micropropagation. And next is production of commercially important plants. Remember, whatever the economically important plants are there, one of the best example, there is a potato plant is there, and tomato plant is there, and next is the apple, right? And the tepu, we know about the wood producing plant, tepu, right? So these are the economically very important plants, or commercially very important plants. See, these plants are produced within the whatever the nurseries we know. Within the nurseries also, these plants are produced because these are having very good commercial value. More number of farmers are required. These plants continuously for the crop production, right? So these are the commercially important plants. Some commercially important plants also produced by the tissue culturing process. And next one is the somatic hybridization. Produces the somatic hybrid plants. how the somatic hybrid plants are produced. We know there is a potato plant cell, and this is one of the tomato plant cell. This is a potato and this is a tomato. And these are the two plants. Whatever that cell walls are there, the cell walls are removed. Whatever that plant cells, cell walls are there, cell walls are removed. This is the only protoplast. This is also only protoplast, right? And these protoplasts are fused by the PEG agent. These protoplasts are fused by the PEG agent. PEG is nothing but polyethylene glycol. Polyethylene glycol. This is one of the PEG agent. The PEG agent is used for the fusion of two cells. The PEG agent is used for the fusion of two cells. Once the two cells are fused, nucleus also fused. In certain cases, nucleus also fused. Once the nucleus, in certain cases, nucleus also not fused. In certain cases, nucleus also fused. Right? Once the nucleus are fused, then this one is forms the single cell. This one is forms the single cell. Then this cell is subjecting for the callus production. This cell is subjecting for the callus production. That callus produces the plants. That callus produces the plants. And these plants are called as tomato plants. These plants are called as tomato plants. Remember, commercially these are also very important plants, right? The tomato plant, lower side means at roots produces the tubers, upper side produces the tomatoes. Upper side produces the tomatoes, right? That is about the somatic hybridization process. Somatic hybrids are produced. Example is the tomato. See, commercially important, but if we compare the original potato taste and this potato taste, somewhat this one is not having that proper taste. The taste is changed, like the tomato taste or the potato taste is changed here. Because the genes are fused here, somewhat taste is also changed. Right? That is about that different applications. And remember, there is a haploid plant production is also takes place. Haploid plants production. So how the haploid plants are produced? Simple. Whatever the anther is there, anther having the pollen grains, pollen grains are subjected for the callus production. Pollen grains are subjected for the callus production. The producing plants are haploid plants. Other way, there is a ovules are there. Okay, ovules are there. Ovules are having embryo sac. Embryo sac containing. Okay, embryo sac containing cells are subjecting for the callus production. Haploid plants are produced. So, by the tissue culture, we can produce the haploid plants also. By the tissue culture, we can produce the haploid plants also. That is about the different applications of the tissue culture.